So welcome students to the second lecture for our course Machine Learning for Health Informatics. Please refer to the course homepage for current information. And don't forget, stay healthy. So what will we learn in this lecture? Before we can or we should do any machine learning, we need a careful understanding of our data. Particularly in the medical domain, information is, as we have learned in the last lecture, the hiatus theoreticus, our bridge between those two worlds, and information quality is what we should aim for good decision making. And finally, we will briefly touch the difficult area of knowledge representation and see how this is done in the medical domain. So data, information, knowledge. But first, we will start with the reflection. Then we will get to know the underlying physics of data. Then where does the data come from? A taxonomy of data, data integration, mapping and fusion. Then information theory, entropy, and finally knowledge representation, a little bit about ontologies and medical classifications, because this is also very important for machine learning. Okay, before we start, we will do a short reflection on the contents of the last lecture. So, kind of a warm-up quiz. So, look at picture number 1, and why do you think is a gap between those two worlds? Or I have even expressed that there is an ocean between those worlds. Why is there a gap between those two worlds? Okay, second question, uncertainty. Why is this a particularly huge problem in medicine? Why is uncertainty for us a big problem? Number 3, why is this seemingly simple mathematical equation so awesome and so important for us? And who was behind the development of it? When you look at this image, number 4, where is the highest certainty in this image and how is the degree of uncertainty described? Quiz image number 5. Why is autonomous driving for us a super practical example for automated or autonomous machine learning? Question number 6. Medical decision making is a search task in arbitrarily high dimensions. Why is the problem bigger the more bedside you go? Number 7. What is the benefit of this human in the loop? Why should we involve a human being in the machine learning pipeline at all? Number 8, why is contextual understanding so important for us? And in which areas are humans, even little children, much better than our best algorithms to date? And finally, number 9, extra question for you. Which important guiding principle of machine learning can be seen here in this picture? And there is a next question, where is the biologist in this image? Correct, as you have guessed, the human wearing the white coat is the computer scientist. The biologist is the machine. So this was described by Pedro Domingos in his book Master Algorithm. Pedro Domingos from the University of Washington, he points out that among the most important issues is understanding the data in the application domain. And data needs a certain knowledge about data integration, about selection of data, about cleaning of data, about not cleaning data, of course, about pre-processing, and of course about interpreting the results. And this is most relevant for the output of our algorithms. I can't mention this often enough to my students. Data and data quality are crucial, and know their data. In machine learning tasks, it is also common that you shuffle data and you normalize it. You can remember our last lecture, we always talked about normalization. And the purpose of normalization is clear. We have some range of feature values. However, Leon Petou from Facebook AI Research, this is the one with the gradient-based learning paper together with Jan Lecan, Joshua Benji and Patrick Hafner from 1998. This is the paper which has nearly 26,000 citations to date. Yes, that is a real influencer. He pointed out at his ICML 2019 keynote last year, nature does not shuffle the data, so we shouldn't either. That means have a look before you shuffle the data. So I always tell my students to look carefully at the data, understand the data, and therefore you need help from a domain expert who knows the background. Otherwise, it will happen like on the next slide. We see on the next slide what happens if you feed in your data into your machine learning pipeline. So you see this guy is asking, this is your machine learning system. Then this guy is answering, yes, you pour the data into this big pile of linear algebra, then collect the answers on the other side. And what if the answers are wrong? This guy is asking. And then this guy is answering, just stir the pile until they start looking right. You might laugh now, but this is unfortunately not funny. This is unfortunately true, because whatever data you fill in and whatever algorithm you use, you always get a result. And the question, or our question in this course, is whether and to what extent the result is relevant. And please remember the word relevant. Relevant for solving your problem or supporting your hypothesis. Yeah, I know what you are saying now. Many people do not have an underlying hypothesis. They do just like in this image. So it might seem funny, but unfortunately it is not funny. So please stay away from just filling in data without any knowledge behind it, what you're doing. Okay, so how to ensure good data quality assessment? So already in 2002, long before this big data hype, Pipino, Lee and Wong wrote this valuable paper on data quality assessment, and their table is really very useful. So you have here just a few dimensions and definitions. I'll just fly over it. You can study it in more detail at home, of course. So let us pick out a few out of them. So for example, start with the first, accessibility, the extent to which data is available or easily and quickly retrievable. Or completeness, for example. Or free of error, the extent to which data is correct and reliable. This is a huge problem in the medical domain. Then look here at this 2002 paper, interpretability, the extent to which data is in an appropriate language, symbol or units, and the definitions are clear. You see, the definitions must be clear. And then, for example, timeliness, the extent to which the data is sufficiently up to date for the task at hand. Also very important. So please study carefully at home. This, if you have a chance, please look up the paper. It's really worthwhile to read. So assessing data quality is an ongoing effort that requires awareness of the fundamental principles underlying the development of subjective and objective data quality metrics. And there is a recent development many years later. The FAIR data principle was launched, and FAIR is an acronym. The acronym describes F for findable, A for accessible, E for interpretable, and R for reusable. Okay, and this is the FAIR data principle. This paper was published, led by Mark Wilkinson, published in Nature. And yeah, I know this is one of the papers where the author list is longer than the content, so just joking. So interoperability, this is a term which is very important, and this is the ability of data or tools from non-cooperating resources to integrate or work together with a minimal effort. And there are some technical solutions for that, but we now will begin with our technical content of the lecture. So this was just as a rehearsal for you, or as a repetition. Okay, so we can summarize what we have heard so far. So data, information, knowledge. Data can be numeric, non-numeric, both. Non-numeric data can include anything from language data to categorical image, video data, and so on. We will now learn about this a little bit more in detail. So what do you see here? Yes, that is an excellent example for independently identically distributed data. This is the famous IID data. This is white noise. And technically, this is a random signal having equal intensity at different frequencies. And this gives a constant power spectral density. And statistically, this is a collection of random variables, all with the same probability distributions as the others, and all are mutually independent. So this is really a cool image. I like that. So this would be a good image for your sitting room. So this is really important for machine learning theory. The IID assumption is often made for training datasets to imply that all samples stem from the same generative process, and that this generative process is assumed to have no memory of past generated samples. So think about that. So we do not have such data in the real world, and we do not have this data, unfortunately, in the medical world. So this is really very important that you look carefully at this image and look about the assumption of identically distributed data, IID data. Okay, so what are then the key problems in medical data science? So the first problem is, and we have mentioned this already last lecture, heterogeneous, distributed, inconsistent data sources, and therefore we need this data integration and fusion. Then we are confronted with complex data, complex meaning high-dimensional data. This is the challenge of dimensionality reduction and the visualization thereof. Then we have noisy, uncertain, missing, dirty, imprecise, imbalanced data. So there's the challenge of pre-processing, but this is a counterbalance. Think about what I have said before. Do not shuffle the data. Look at the original data on the one hand. And then I'm saying here, you should pre-process the data. So if you pre-process, you shuffle, you alter the data, so it's no longer the original data. So you see, this is a trade-off. And students, please, if you are a good data scientist, then you have to carefully check by yourselves to what extent do you have to look at this data, and you need always the opinion of the expert. That means the medical expert knows what is going on in the data, the medical domain expert. So you need this integration of the medical domain knowledge. So then the discrepancy between data, information, and knowledge, there are various definitions. Also in this course we will have some definitions and we will follow these definitions. And then what is called big data sets in high dimensions. So this manual handling of the data is often really impossible. So this is just large data. amounts of large data sets. So, why can data in machine learning often not be represented by a simple model? So, in their famous Nature paper, the Turing Award winners Lecan, Bengio, Hinton, you will hear these names quite often in machine learning, they three emphasize how important the focus on data is for machine learning and what the typical differences are when looking at data from traditional statistics and from the viewpoint of modern machine learning. So you see, you have also here two worlds. You have the statistical, the traditional classical statistics, and you have this kind of modern machine learning. So data in traditional statistics is low-dimensional data, often less than R of 1 to the power of 100. The problem, you have always much noise in the data, but not much structure in the data, but it can often be represented by a simple model. And a simple model, for example, is linear regression. Linear regression is a good example for parametric, being parametric. That means if you have a finite and fixed number of theta parameters, remember the theta parameters are our features, and this is regardless of the data size. So we can call this model-based learning. So this is relatively easy. On the other hand, you have data in machine learning. You have often very high dimensional data, so much greater than R to the power of 100, much greater, to 1 million or even 10 million. The problem is not the noise, but this complexity, so it is just a lot. And there is much structure, but the structure cannot be represented by a simple model. And this is our problem. For example, neural networks, they are highly nonlinear, they are non-parametric. So the features may grow with the data size, and we call this often, because of this, we call this often model-free learning. And exactly this makes it difficult for us. So this is a very nice slide where the difference of traditional statistics and machine learning comes out very clearly. So why is the curse of dimensionality for us relevant? So here you see the famous paper of the Bengio brothers. This is Sami Bengio and Joshua Bengio. And this was described in 2000. And you see 1 dimensions, you have 10 positions. 2 dimensions, you have already 100 positions. In 3 dimensions, you have 1,000 positions, possible positions, and this goes on with dimensionality in the mathematical way. So the curse, Fluch, das ist in German Fluch, the curse of dimensionality hits particularly hard on models of high-dimensional discrete data. This is exactly our neural networks. And there are many more possible combinations of the values of the variables that can possibly be observed by a human, for example. Yeah? And this is also an old idea. Originally, this goes back to Richard Bellman in 1953 when considering problems in dynamic programming. So basically on various phenomena that arise when analyzing and organizing data in high-dimensional spaces. So you know often we have these hundreds or thousands of dimensions in the medical data domain, and that do not occur in low-dimensional settings, such as in the 3-dimensional physical space of our everyday experience. So you have to discriminate carefully between this physical space where we live in and the mathematical space, which can have arbitrarily high-dimensional spaces. Okay, what is the difference now between data, information, and knowledge? So there's often confusion between the terms data, information, knowledge. And to clear this up from the beginning, for this course, we will follow this model, which is based on Atkinson, Schifrin, the Wickens model, but I will call this my own model. Okay, so we have this outside world, where we have this internal world, so the outside world, the internal world. We have 3 filters. the last is called mental models. And then we have these stimuli. So we have this ongoing physical sensory input. We call this stimuli from outside. So this is the ordered sequence of data. And then we have this selective attention. I have mentioned this last time. And due to selective attention, we perceive only a fraction of this data into our second memory system, in the second filter, and then only a fraction goes into the central or the long-term memory, where our mental models are there and where we check if the incoming perceived data is fitting into the information which we have stored as previous hypotheses. So this is also a good example for this previous knowledge. So then we have feedback at all levels, and of course the feedback to the outside world, although we have here again this selective filter. And then we call this stimuli from outside the data. Then we have this transforming of data into information, and then we have knowledge. And now this is let's say the important part which you should remember now. For us, knowledge is a set of expectations, and this is the terminus we will use in this course. So this fits nicely into the definition of the informatics professionals, the American Medical Informatics Association, the AMIA. And I like the definition of the American Medical Informatics Association. And this was actually written by Edward Shortleaf into our 2011 volume on information quality, where we had the first international conference on this subject in Graz. So when Edward Shortleaf and Vimla Patel were our guests. And I should mention that Edward Shortleaf is among the early pioneers of health AI, and he is the father of MyCin. This is maybe the most famous expert system and one of the early pioneers what is now called explainable AI. So just a side note, we will learn about expert systems later on. So what does the professionals say? So biomedical informatics is the interdisciplinary field that studies and pursues the effective use of biomedical data, information and knowledge for scientific problem solving and decision making, motivated by efforts to improve human health. So this is a beautiful sentence. You have in exactly the content of this lecture now: data, information, knowledge for the purpose of problem solving and decision making, and of course to improve human health. So this is really a very, very good definition, and we will follow this American definition. So where do the data come from, and what types of data do we have in health? By the way, this is John von Neumann and this high-speed computer in approximately 1952. So our question is now, where does the data come from and what kind is it? So how can we classify the traditional data structures? So you know we are speaking always about multivariate data set. This is a data set that has many dependent variables, and they might be correlated to each other to varying degrees. And usually multivariate data is with discrete data, and then we have multidimensional data set. This is that has many independent variables clearly identified and one or more dependent variables associated to them, and usually we call this, or it is associated with continuous data models. So just remember: multivariate, discrete, multidimensional, continuous. So yeah, you know, in other words, every data item or object or physical stimuli of our world in a computer is represented or stored as a set of features. And instead of the term features, we may use the term dimensions. and because an object with n features can also be represented as a multidimensional point in an n-dimensional space. So this is dimensionality reduction when the process of mapping an n-dimensional point into a lower k-dimensional space, and you will see this later on. This is the main challenge in visualization of our machine learning pipeline. So the number of dimensions, we will see this later on, can be sometimes small. For example, simple one-dimensional data, such as a temperature measurement at different times, and going on to high-dimensional data. Okay, but now how can we classify traditional data structures? You see this in this slide. So we have aggregated attributes here and separable. We will concentrate on the separable. And here we have qualitative and quantitative. And this is important for us because with this we are dealing most of the time. So you see qualitative, nominal, ordinal, quantitative, interval, and ratio. And now this is very important to go back to the early origins of this. So what was the original work on the theory of scales claiming? So this is the original work of Stanley Stevens from 1946 in Science, where he claimed that all measurement in science was conducted using these 4 different types of scales, and he called it originally nominal, ordinal, interval, and ratio, unifying both the qualitative, which we have seen in the last slide, which is described by his nominal type, and quantitative to different degree, all of the rest of his scales. So the claim was that all measurement in science was conducted or can be conducted by those 4 types, and therefore I'm mentioning it because this is really important, and therefore let's have a closer look on that. So this is really now the essence of data. So we have the properties. So what properties do separable data have? So we have this in terms of scale, empirical operation, the mathematical group structure, the transformation possibilities in R, the basic statistics we can do, and the mathematical operations that can be performed. So you see this is the nominal, and you can check this out at peace at your home later on. So please carefully study this. The ordinal, the interval, and the ratio. And now you have a nice slide where you have all this data in one good overview. And now go back to the more physical nature of data. So what levels of data taxonomy can we identify? So of course, most of our computers are von Neumann machines, digital computers, and consequently at the lowest physical layer the data is represented as patterns of electrical on-off states, 1, 0, high, low, blah blah blah. We speak of a bit, and bit actually is binary digit or Shannon-coded basic indissoluble unit. And this is not equivalent to a bit with the capital letter. And in quantum systems we are talking about a qubit, for example. So regardless of these physical realizations, for example, with voltage or with the mechanical state or black and white, it is always a logically 0 or 1. And this is quite analog to a light switch. And the qubit, this is in quantum systems, has similarities to the classical bit, but overall it is very different. So please be careful. A classical bit is a scalar variable with a single value of either 0 or 1. So the value is unique, deterministic, and unambiguous. This is important. While a qubit is more general in the sense that it represents a state defined by a pair of complex numbers, so A or B. And this expresses the probability that a reading of the value of the qubit will give a value of 0 or 1. And thus a qubit can be in a state of 0 or 1 or some mixture. And this is referred to as the superposition. And therefore, please be aware that this model of quantum computation is not the only one. So quantum computing is something different. So please, I just mentioned this for the sake of completeness, that this is a different system. Okay, so we continue with our levels of taxonomy. So then is the logical level. So the logical level, we have primitive data types, including, for example, Boolean, true, false, or numerical, for example, integer, set, for example, floating point numbers, reals, etc. Whereas we don't know what is really a real. Then we have alphanumeric strings, for example, and then we have conceptual abstract level. So these are our classical data structures as we have learned it in informatics. For example, lists, arrays, trees, graphs, and so on. So this is the abstract level. And then we have the technical level. This is the application data format. So for example, a text or graphics or images, pixel for example, or vector, audio, video, multimedia, and so on. And then we have the hospital level, and this is also a really important level of data. So in the hospital level, you have a lot of narrative data, textual, and we will come back to the importance of text later on. Then you have numerical measurements, for example, coming from physiological data, from lab results, vital signs. Think on the intensive care units we have seen last time. They generate constant numerical measurements, a lot of them. Then you have recorded signals, for example, EEG, ENG, EMG, EOG. There's dozens of them. And then you have all the images, for example, like coming from cameras, simple cameras, for example, operation cameras, then X-ray devices or magnetic resonance, computer tomography or PET devices, MRI, fMRI, and so on. And then, of course, you have the vast area of omics data, and we will spend some time for this omics data because they are now really very, very present. And you see at the first glance, at this simple slide, you see already the complexity of the hospital level and the different and the variety of different data, and that is not easy to now to integrate all this data, to fuse all this data together. And for one reason, think of this to produce quality information for better decision making. So at the end of the day, somebody, the medical doctor, has to make a decision based on the information given. And this information is created from this data. And this is a vast area in the medical domain. Okay, so where do data come from at hospital levels? So you have, of course, clinical workplace data sources here, medical documents, and here I have to point out text is important. We will have a closer look on text. And text is often called, not quite correctly, free text. And the correct terminus is non-standardized text, because we have standardized text and we have non-standardized text, and we will come back to this later on. But it can also be semi-structured. You can use standard terminologies, and we will get to know some of them in this lecture today. So ICD, for example, or SNOMED CD. And then we have measurements, for example, from the lab, a lot of time series data from measurement devices and so on. Then you have, of course, a lot of survey data, clinical study data, trial data, and so on. So these are the clinical workplace data sources. Then you have image data sources, just that you have an idea. This is radiology. So radiology produces a lot of data with a lot of different modalities. We call it modalities in radiology. I will show it to you a little bit later on. Then, of course, we have digital microscopy, digital pathology. We also will discuss this a little bit in more detail later on. Then, for example, confocal laser scanning is one device which is used often in dermatology. And then, of course, you have these vast resources of omics data. There are so many different omics meanwhile. So these gene sequencings, and these are really a lot of data of various size and various difficulty. And about this later on, just that you have an idea where do data come from at the hospital level. Okay, so what are typical examples of imaging data? So radiology, of course, is the standard, going back to Konrad Wilhelm Röntgen. So this actually Konrad Wilhelm Röntgen received the Nobel Prize in Physics in 1901. Here you see the typical images like this. So this is a colon. This is clearly seen on the air contrast barium enema here, for example. So the white areas are barium, this is the contrast, and the black regions are then the air here, for example. And the contrast agents are used to make organs in the body visible on the images, otherwise you wouldn't see that. And they can be given by injection into the blood systems or via tubes into internal organs. Yeah, and here on number 2, a typical X-ray radiograph of the chest, in which the regions of bone appear white, for example, here, you see that. And then figure 3, you have a normal breast tissue, and it appears on a mammogram, digital mammography. This is also known as full-field digital mammography, FFDM. This uses electronic detectors that convert X-rays into electrical signals, and the images of the breast can be viewed then on a computer screen. So advanced developments in mammography enable considerable reduction in the required radiation dose, because this is an issue here. With all these X-ray images, you have always the danger of radiation. And new software is available that can further reduce the dose up by many percents while maintaining the same image quality. So this was not the case in earlier times. And then figure 4 here, angio, cardiac imaging acquisition using a special low dose program, for example. Then number 5 is a typical PET or CT. This exposes the patient to a small amount of ionizing radiation, and you see this can be colored. And this is been seen in more detail here. And number 6 is a PET and MRI imaging device, and they are now required to resolve diagnostic questions. And they have a key benefit: the 2 examinations can be performed in one single scan. So this is state of the art of radiological imaging. So why is digital pathology interesting? So this is one of the most promising fields of diagnostic medicine, and digital pathology is not just the transfer of histopathological slides into a digital representation. It is much more. This is actually a very good example for data integration in the life sciences, because the combination of different data sources, for example images, patient records and omics data, together with current advances in AI, for example machine learning, enables to make novel information accessible and quantifiable to a human expert, which would not otherwise available and not exploited in current medical systems. So what you see here in the image is a typical digital pathology workflow. So here we start with the patient. We have the primary sample here, and then we have these tissue blocks, and then we have these slides here. And then we have this DNA, RNA sequence data. Then you have the data stored. You have the virtual microscope with the pathologist, and the pathologist is producing the reports here. And then you can, for example, integrate the data from the bioinformatician, for example, with the genomic data, with the omics data. So the grand goal is to reach a level of usable intelligence where you can fit together the data together, so that a kind of an augmented pathologist can make the best possible decision out of the available data. So how is such a whole slide images WSI produced? So here you see this is such a slide, And for you as information, histopathology is the study of how tissues change during disease. So this is a rather gold standard for disease diagnosis. And here everything starts with the real case. So you take the tissue samples during, for example, an ongoing operation or a biopsy. And here you can see a schematic view on a histopathological slide. So you see here the area, the target area is the de facto standard for quoting scan speed and size. And with help of a scanner, with the whole slide imaging scanner, like you see here, the histopathological slide is transformed into an electronic image. So this is a virtual slide. This is called whole slide image. And this is a digitized histopathology class slide. This had been originally created on a slide scanner and represents now a high resolution volume data cube, which can be handled then with a virtual microscope, and most of all where we can apply all sorts of AI and machine learning approaches here. So this is the slide here. And here in the right bottom of this slide, you see the detail of the result. So this is a typically stained histological section of a formalin fixed and paraffin embedded normal human liver tissue. So this is just a tissue of a human liver. Yeah, and what is the current state of the art in machine learning here? So whole slide imaging is becoming a routine diagnostic method. So this accelerates, of course, the clinical diagnosis from the pathology images, and it automates image analysis in an efficient way. And here also you see deep learning is applied. So this is basically image analysis with deep learning approaches. So you can have a look at this paper, for example, that you have a state-of-the-art insight into how this is currently done. So what about the ground truth? This is the main question. The ground truth is coming from the pathologist, you see. Therefore, this is of utmost importance, and this is a nice example of the difference between data of the pathology image, then the information you produce for the decision making, and you need the knowledge of the pathologist as a ground truth. A good example is neonatal screening for data generation. So this is now another source of data. This is done in Austria the standard basis. So every baby is taken a Fersenblut. This is Hilbprick. And this blood is actually a good example for valuable data. And this is done in such machines, in metabolite machines. And you can do a lot of this, and you can find out a lot from the blood, for example. So blood actually, and this is the sense of this slide for you now, blood is a really good example for a really good data set where you can find out a lot of, where you can gain a lot of information. So what is an example for the data structure list? So in biomedical informatics, we have a lot to do with such abstract data types, and here just one important one. So this is the list. List is a sequential collection of items. You see this here. And the typical list here is a combination of DNA sequence. And these are nucleotides, and these are joint molecules which form a structural unit, and these play a central role in metabolism. So this is just a data structure list. Then graph, and with graphs we will deal in a later lecture. So just what we mentioned it here, graph-based machine learning is maybe the hottest topic at the moment, and we will spend the whole lecture on it. So graph is a pair G of V and E, where V of G is a set of finite non-empty vertices. These are called the nodes. You see here the nodes. And EG is the set of edges. These are the lines, and this can be two-element subsets of VE. So this can be ordered or non-ordered, directed edges or errors here. Then it is a directed graph or it is a non-directed graph. And this, the good thing is, it can be represented as an array in a matrix, and then it can be really nicely calculated, because this is good when there are zeros here, you know, then you save a lot of computational power. So what is a tree? A tree is a collection of elements called the nodes, one of which is distinguished as a root node along with the relation. This is the parenthood that places a hierarchical structure on the nodes. And a node, like an element of a list, can be whatever type we wish. So we often depict a node as a letter, for example, a string or a number with a circle around it. So this actually, and this I should mention, is also called dendrogram. Dendrogram from Greek dendron, tree, and gramma, drawing, so tree drawing. And this tree diagram frequently is used to illustrate the arrangement of clusters produced by hierarchical structuring and clustering. And this is often used in computational biology to illustrate the clustering of genes, for example, like here. And the origin of this is Darwin. So actually Darwin is the founder of this dendrogram, Darwin 1859. Okay, so we can summarize what we have learned so far about data. So remember, we have different data, from structured, categorical data, semi-structured, and so on, to total unstructured. We can have narrative, free text, and so on. And now let's go into more medical terms. In medical informatics, we have the permanent interaction between different data, and what are the origins of health-related data. So here, this figure is from a highly cited paper of us and demonstrates the extreme broad origin of health-relevant data. So this is really very challenging. So if we start here at the bottom from very microscopic dimensions on an atomic level, so typical example is X-ray crystallography data, as you can see here, for example. Then you have molecules, proteins, lipids, DNR, RNA. At the moment, in every news, it's a certain virus, COVID-19, for example. So we have these positive RNA viruses. They account for a number of known viruses, including the SARS and MERS and coronaviruses and SARS-CoV-2. This is the most recent one. But this is also of similar size than the typical influenza or rhinoviruses for Schnupfen, for example, which are omnipresent in mankind. So remember that there are about 5,500 viruses known so far, and there are a lot of unknowns. So then we have the much larger bacteria up to the cell level, fat cell, blood cells. We see a beautiful slide a little bit later on. Then we have the tissue of the individual human, which we will also examine on the next slide. Then the tissue forms organs, for example, hearts, lungs, the brain, of course. And fascinating for me is always what makes a cell to know that if it is working as a brain cell or a liver cell. So then, of course, there's the individual human here. And then we have the collective. There's a bunch of humans, a population, and then we have a whole ecosystem. And at the moment you see, like infection data in pandemics, they affect a whole ecosystem worldwide. So this is a very good example at the moment. And you see data. This is everything of data, and you see how fascinating it is and how many dimensions you have to cover here, from very tiny to the very large macroscopic dimensions. So because there is much different data here, just some examples. So why is data integration, health, and unsolved problems? So just that you have an idea. So we have, if we start here maybe with microbiome, so this is a microorganism, processes, plants, fungi around your body, or you have internal metabolomics, chemical processes, then you have a lot of outside data, electronic health records, for example. Then relatively new is the field of exposomes. This is all about environmental data, for example, air pollution, exposure of toxicants affecting a human body, which can be measured by sensors, for example. Then we have this collective data, social data, fitness, wellness, ambient assisted living, and so on. Then foodomics, lipidomics, we will see omics a little bit later on. We have discussed this imaging data, X-ray, ultrasound, and so on. But there are a lot of other video data, for example, from observations, for example, from the sleep laboratory, or from gait, from child walking, for example. And then, of course, transcriptomics, RNA, microRNA, and so on. And genomics, epigenetics, proteomics, protein-protein interaction. So you see data integration is really an unsolved problem because it's so much and so complex. So why is the human exposome important? So we have recently launched a new European project on the exposome, and the exposome is intriguing because it encompasses a set of human environmental, that means non-genetic exposures from conception onwards, so complementing the genome. So exposome was first proposed in 2005 by cancer epidemiologist Christopher Paul Wild in an article entitled Complementing the Genome, and with an exposome. And the outstanding challenge of environmental exposome measurement is in molecular epidemiology. So the concept of the exposome is to assess everything which comes from internal, from external effects, so also including social capital, education, financial status, psychological stress, like we experience now at the moment, so bound at home, so it's not so funny. And then, of course, the urban, rural environments, the climate, for example. So it's a huge difference if you live in the polar region or you live in the equatorial area exposed to big temperature changes and so on. And then, of course, specific external, for example, radiation, think on the nuclear things, or the Feinstaub, then infectious agents. At the moment, for example, these viruses, influences of this virus, but also part with psychological stress. So this is actually a very good example at the moment, this corona pandemic. This influences you from this external and specific external, and also to the internal world. So this is really a very good example for this. And and this produces a lot of data because sensors are measuring this data constant. So you can measure everything with these sensors, and radiations, for example, but also, for example, physical activity, and of course all sorts of internal data. Just that you have an idea about this data source. So what is a good example for the level cell? The cell is actually the most important, and I showed this per intention to you because this is awesome. First of all, life is information. So this goes all back beyond pure data science. So this is information, and this reminds us on the famous paper of Schrödinger, which I show you later on. And cell comes from Latin cella, meaning small room. And this is the basic structural, functional, and biological unit of all known organisms. So a cell is the smallest unit of life. And therefore cells call as building blocks of life. And the study of cells is called cell biology, cellular biology, or cytology. So just that you have an idea. And the awesome fact is that the human body, so your body, contains approximate 4 times the 10 to the power of 13 cells. So and the brain accounts for around 80 in early times. We estimate 100 billions out of this. So, and just for the sake of completeness, you have to discriminate between two types of cells. So we have eukaryotes, plants, animals, humans, where the DNA resides in the nucleus, and we have these prokaryotes, bacteria, which do not contain compartments. But this is very interesting, a good example for life. Life is information. So, now this famous acclamation of Schrödinger. So Schrödinger's early ideas encouraged many scientists to investigate the molecular basis of life. And actually Schrödinger stated that information is negative entropy and is the abstract concept that quantifies the notion of disorder among the building blocks of life. So what is life according to Erwin Schrödinger? It is something which is reproducing, or to reproduce, to grow, to evolve, Darwin, to self-replicate, to generate, utilize energy, very awesome, and to process information. So life is a fantastic interplay of matter, energy, and information. And therefore I'm showing it to you, because in this course, in this lecture, we are dealing about data, information, and knowledge. And now we are deep into information. So scientists in artificial intelligence and artificial life are interested in understanding the properties of living organisms to build artificial systems that exhibit these properties for useful purposes. So AI researchers are interested mostly in perception, of course, in cognition, and in the generation of action, whereas artificial life focuses on evolution, on reproduction, on the morphogenesis, and on the metabolism. So this is really, really very interesting and very awesome, and goes back to Erwin Schrödinger in 1944, What is Life? So just that you have a pointer to some open data sets. So there are billions of biological data sets openly available. So we are living in an open data world. So there are a lot of data, because the question now would be obvious: where do you get the data from? You can retrieve data from a lot of open source around the world. Here just one example, so very tiny examples, just that you have a pointer to some open data sets. Okay, so I hit the URL, so it moved forward. Okay, next slide. What is the most important, what are most important omics data? So there are a lot of omics data generated, and here, just for the sake of an overview, the most important one. So of course genomics is sequence annotation, then transcriptomics from microarrays, proteomics in the proteome databases, for example, metabolomics, this is enzyme annotation data, fluxomics, all about isotopic tracing, metabolic pathways, phenomics, for example, biomarkers, then epigenomics, this is about epigenetic modifications, microbiomics, microorganisms around our body, and lipidomics, these are the pathway of cellular lipids. And this is just a fraction of all of them, but just that you have an overview. The next slide is a little bit more difficult, but this is for study if you have time. This is a little bit about omics data integration. So omics data integration helps us to address interesting biological questions on the biological systems, and this goes towards personalized medicine. I will explain this a little bit later on, but a major challenge in our networked world today is this increasing amount of data, and you know, all of this big data. So actually in the medical science we have this big data, and so here a little bit an overview about different genomics, transcriptomics, proteomics, metabolomics, protein-DNA interactions, protein-protein interactions, and fluxomics and phenomics. So just that you have an idea and an overview of it. So, examples for lower dimensional data. So far we have spoken about this high dimensional data, back to some physical diversities in engineering, physics and so on. So we have the variable property of a subject. So for example, zero dimensional data is just a data point existing isolated from other data. These are also integers, letters, booleans, for example. Then we have this one-dimensional data consists of a string of zero-dimensional data, for example, sequences representing nucleotide bases and amino acids. Smiles is a typical example. Then we have this two-dimensional data. These have this spatial component, for example, images. Then there are 2.5-dimensional data. I will give you later an example of it. This can be stored as a 2D matrix, but can represent biological entities in 3 or more dimensions. This is the famous Swiss PDB records, the protein database records. This is awesome. You can play around with this. So this is really very interesting. I will show this later on. And then we have this real 3D data. These are voxel data. So this is the density maps. And then, of course, we have all this high dimension data, which we cannot physically visualize here. Okay, so what is a good example for one-dimensional data? Univariate sequential data objects. So SMILES. SMILES is short for Simplified Molecular Input Line Entry Specification. This sounds difficult, but this is just a compact machine and human-readable chemical nomenclature. For example, Viagra has this nomenclature, and this can be disposed here and visualized. And this is well documented. So you can see this here. Then we have a typical example for two-dimensional data, bivariate data. So this is the proteomic analysis of men's enzymal stem cells. So you get this two-dimensional Gale electrophoresis on using this whole protein cell, and you extract this and then you get this data. So this is typical proteomic analysis to get from a two-dimensional electrophoresis. Yeah, and this is the 2.5 data, structural information and metadata example from the protein database. This is very awesome. You can see here a vast amount of protein interaction data, and this is just beautiful. You can look this up. Yeah, and then what are 3D voxel data? These are volumetric picture elements produced by medical imaging devices. This is transaxial left, coronal middle, and sagittal right. This is a patient who was scanned for 30 minutes in list mode with the brain PET scanner. And this is a typical voxel image. Okay, now, so what can we say about data integration, data mapping, data fusion now? So what do we mean with data integration? And I show you this old. This is a work from Kirsten et al. From 2006. This is an old but very nice example what we mean with data integration. So here you see these clinical centers, pathological centers, genetic centers. So you see these different modalities here, clinical findings, text data, for example, pathological findings, images, for example, histopathological images, and then genetic center. This is all the genetic data, and all this has to be fused together to validate it, and then to get out for a study and for basic reports, so about decision making. And this is a nice example about fusing together different data for making a joint decision. So this is a collaborative clinical research study. So what is our goal of data integration? So the goal is to get a unified view for decision support. Please remember, we have to find out always what is relevant. And this is the real big problem, to determine what is relevant and what is not relevant. And what is not relevant, you should ignore. And what is relevant, you should take on for decision making. But this is hard. And this integrative analysis, as it is pointed out here, this integrative analysis requires a systematic combination of various data sets and diverse algorithms also. and this is support of multiple user needs and a unified view, and therefore multi-view learning is so essential. And not everything what is interesting is also relevant, so the essence of our profession is to provide the relevant information. So you see this here. Yeah, this is a famous paper from by Joseph, so distributed information processing in biological and computational systems, and the main essence is how to combine these different data types together to obtain a unified view of the activity in the cell is one of the major challenges of systems biology, and of much more, of course. And this brings us back to the slide we have seen in the last lecture. So why may information bridge the gap between two worlds? So information, you see, can actually really bring together, because this is what both needs, this joint information, this quality information. This can be the hiatus theoreticus for decision making, because this is what both needs, both sides. So this can be the bridge then. And why is imaging data alone not enough? So this is a good practical example is precision medicine or P4 medicine. So now it's the point where I have to explain what is precision medicine or personalized medicine. So let's start maybe with some explanation what is traditional general medicine. So general medicine sees the human as one living species and develops drugs or treatments and therapies which should cure each and everybody. So we have a muster patient, and this is a typical patient. This is 75 kilograms, male with 107 centimeter height. So far so good. We all share the generalization that we have underlying biological principles. So biologically this is quite true. And to a certain extent, each of us is different, has a different individual and personal background. And this is unique, or it is at least shared by a smaller group of humans, for example, women or elderly or diabetes patients, etc. So the first idea is in stratified medicine. Stratified medicine would mean to bring certain groups together, for example, women, or elderly people, or diabetes patients, and so on, so to form a stratified group. And personalized medicine would be a grand goal. And personalized medicine, the better term is precision medicine, because actually this is describing what is really done. So precision medicine is a medical model that separates people regarding medical decision, practice, intervention, and or products being tailored to the individual patients. So based on their predicted responses or risk of disease. And this needs, of course, a total data state of the art, so that you have a good overview what is the state of the art, and then you can tailor-made drugs, for example, individually, personalized and precision. And the official term is P4 medicine: predictive, preventive, precision, and personal. And for us now it is important to know that for a good decision making, imaging data is not far enough, and this is the output of this paper. So for a good decision, imaging data is not enough. So if you look at the P4 pipeline here, you can see how much more contributes to a treatment tailored to individual patients. You see these omics profiles across genome, proteome, metabolome. Then we have these network relationships, these link relevant entities which in each data layer. Then we have these layers of annotated networks, annotated with tissue disease, network properties, and so on. Then we have these discovered relationships across data layers, identified combined biomarkers, and so on. Then combined biomarkers identify clinically relevant patient subgroups. And then finally, a treatment tailored to patient subgroups results in improved patient outcomes. So, and the ultimate goal would then be the treatment tailored to an individual patient. You are an individual, you are unique, and you get the personal precision track exactly to the needs what you have. So, and this works then with you only. Okay, so this is the idea of personalized preventive precision and predictive medicine. But of course, this is a high ground goal, and this is, let's say, also a cost question, of course, because you can imagine that this is very costly, of course. So, what is then translational health? So, I have to mention this translational health or translational medicine, so often referred to as translational science. So, this is defined by the European Society for Translational Medicine as an interdisciplinary branch of the biomedical field, supported by three main pillars, and you can see this here. This is the bench, the laboratory, the bedside, and the community. And the goal of this translational medicine is to combine these disciplines, these resources, this expertise, and also the techniques within these three pillars to promote enhancement in the prevention of illnesses, in the better suitable diagnosis, and in better individual therapies. And this is also let's say, an approach to a synergistic relationship across this biomedical information and translational medicine continuum. So just that you have seen this. And here again, these molecules, cells, tissues, organs, individual and populations level. You see, this is health policy. And now it's beautifully seen also with this corona pandemic, how this works together from bench, where scientists are looking for treatments, bedside, where the doctors are currently treating the patients, community and policy, for example, public health, informatics, populations. How can we secure and make safe our population? Okay, so what are the key problems then in this medical data management? This is just described by heterogeneous data sources. This is a natural need for data fusion, data integration, then of course complexity, noise, the discrepancy between data, information, knowledge, and big data sets where manual handling is often not possible. So among all these different data sets, we can locate 4 different areas. For example, this biomedical research and development data, for example, clinical trial data. Then we have the clinical patient data, for example, the electronic patient record, the lab, the reports, and so on. And then we have this private patient data from ambient assisted living, monitoring, but also this is also including now wellness data, for example. And then we have the health business data, for example, costs, utilization, etc. And among all these different data sets, there is one very important combining link, and this is text. And text is unstructured information, not unstructured data. Unstructured data is something different. Please be aware of that. It is unstructured information, so unstructured information, non-standardized text. So why is medical text important? If you look at the image, the medical record here on the right hand side, this is the text, this is legally binding. So there's a proverb, a long debate, is a picture worth 1,000 words? And the answer clearly is it depends. But of course, in the medical domain, you need the medical record, you need the text, because this is legally binding. So this brings us now to a digression about medical communication. Digression, Umwege erhöhen die Ortskenntnisse. So, why is medical work relying on team communication? So the teamwork in the hospital requires a lot of communication and information exchange. So please remember now how important it is that we speak about information. So it's not about data exchange, it's now about information exchange, okay? And the vision of a business enterprise hospital information system is to cover all the workflows and organizational processes and information flows electronically. So the work of physicians is heavily influenced by the usability of the available equipment, of course, but Please remember now, decision making is never done alone in the medical domain. It's always a concerted effort of many participating people. And the medical report is the documentation of this process. And this goes actually back to Hippocrates. This is very interesting. So what are the problems of the medical report? So as I have aforementioned, the goal is an all digital hospital, produces a lot of data, but most relevant is the unstructured text, and this is the Arztbrief. And here you see such an example, a medical report of a medical image, and this report is very important because it's legally binding, but it's difficult because it contains a lot of special words, particularly German words, a mix of different words of Latin and German and so on. A lot of abbreviations, and this is a big problem and brings us back to the question of ground truths. The abbreviations are often locally different, so every doctor can invent his own abbreviations, and there are some local specifics, and we are far away from this international standardization. And then, of course, errors. So incidentally, there are, of course, many errors in this, and this is a very huge problems in the medical domain. So this handling with this unstructured data is a mega challenge and brings along a lot of challenges for our computers. So this is not a joke. This is a standard case. So this is it should not be funny. So this is a standard German report, and you see this is difficult for us, but you can imagine how difficult this is for your deep learning algorithm, okay? A lot of mixture between Latin, local abbreviations, and so on. So, for example, also this synonymity. So synonymity, ambiguity is a huge problem. For example, die Antrum-Schleimhaut ist durch Lymphozyten infiltriert, but it's the same as lymphocytäre Infiltration der Antrum-Mucosa, absolutely same, or Lymphozyteninfiltration der Magenschleimhaut in Antrum-Bereich. You see, this is totally different sentences, but meaning exactly the same. So this is a big problem, the synonymity and ambiguity of this language. So now this brings us back to some more basic questions. So why does language understanding require knowledge? So of course we have the syntax in the set of rules, principles and processes. We have at the beginning we have the syntax, then we have the semantics, the Bedeutung of these words, then we have the pragmatics. That means pragmatics means the conversational implication, so the interaction with others. This is the practical usage of the language, and then we have this context, and I have mentioned context many times, and I will mention this many times because this is most important, and we are far from our machines can understand context, and such misclassifications, for example, may seem funny but can bring serious trouble. And of course we are far away from emotions. So emotions, this is purely human. We have a certain gut feeling. You can solve often problems where you have not the foggiest idea what is about, but you have you make the right and proper decision, so, and you cannot explain it. So this is also very interesting. And of course our machine learning algorithms are far away from doing that. So they can beautifully work on syntax, maybe also on semantics, but difficult with pragmatics and cannot do the context part. So here this is a very interesting picture from Stanford. This is from Andrei Karpathy and Fei Li. There are a lot of such pictures on their homepage. Please have a look at it. Might seem funny, but the young boys holding a baseball bat, this can make serious trouble, because if a police camera is detecting this, so maybe they can send a police car to them. So this is completely misinterpreting, and this is just a good example. So we are far away from this contextual understanding of images or of any data. So why is text a good example for non-standardized data? Because text is in the core of linguistic data. It contains speech sounds, phonemes, words, phrases, sentences, and the literal meaning of phrases and sentences, and then the meaning in context of a discourse. Okay, and this is called the pragmatics, and this is not standardized at all, because we can use words in completely different meanings, and this does not even include emotional expression. So this is really very, very difficult for computers. Okay, so what are then the typical challenges in data-driven medicine? So increasing large data sets due to this data-driven medicine. Then increasing amounts of non-standardized data and unstructured information, salopply spoken freitext or free text. Then we have heard now about this, data quality, big problem, data integration, universal access to the data. Then privacy, security, safety, data protection, data ownership, fair use of data. Many unsolved problems in here. Time aspects often ignored. Medical diseases, and now also this COVID pandemic is a very good example about the development of a disease over time, and you see how important time is, and time is often ignored in data, and therefore time aspects are of utmost importance. So this is really very essential, and this means a further digression. Data augmentation. So data augmentation will be your first assignment. This will be done by Markus Blois. He is preparing a task for you in Python. This will be your first Python word, and data augmentation is a really very important now, merely state of the art in deep learning process, and I will explain quickly why. So what is data augmentation? So this is basically generation of artificial data via expansion of your own data set. You will now ask why? Because these neural networks require big data. So augmentation is now basically part of most of all deep learning projects. So this is standard issue now. Then it is also used to address issues with class imbalance. So class imbalance is a huge problem. It is cheap, relatively easy to get more data, which will almost certainly improve the accuracy of the trained model. So more data is always better data. Then it improves model generalization, model accuracy, and can control overfitting, okay? Because then you have a suited amount of data. And class imbalance, maybe I should mention what is class imbalance. Maybe you have heard it. Class imbalance occurs in classification problems where you have one class with very few examples compared to other classes in your data set with very huge examples. So in a binary classification problem where class A has maybe 100 samples and class B has 10,000 samples or even more, and neural networks have a real difficulty in learning good unbiased models using datasets with high class imbalance. And this relates to how neural network weights are updated when learning. And as I have mentioned, it is cheap now and relatively easy to get more data that can make a model more robust to unseen data. So in many situations is data augmentation just a no-brainer. So this is just a need. So image augmentation is most common because text augmentation is much harder, and deep learning is applied to images, but it comes now to more and more text. So we will also touch this text augmentation problem. And how is it done? By making label-preserving transformation to the original images, for example. So you can rotate, you can zoom in, crop, and so on, and you can produce a lot of more images. How is it done here? How does this image augmentation work? So, of course, augmentation can be more advanced than just doing some rotations and translations through this axis. For example, here in this image, we have random elastic distortions where the center of an image is distorted without affecting the size or aspect ratio of the image. And on our GitHub page, you will find both Python and Julia versions of our image augmentor. And your first assignment will be on these augmentations. But now back to our general lecture topic of the day, data information, and now about information and a little bit about information theory, and this will bring us quickly to entropy. So why are probabilistic models so important for us in medicine? So you have these Boolean models, these set theoretic models, and these algebraic models, for example, vector space, for example, latent semantic indexing is a good example for that. And you have these probabilistic models, and this is very, very important. And because this is so important, I will now read one-to-one from this slide. So this is originally from Zubin Garamani from Cambridge. So our probabilistic models describes data which we can observe from our environment. And if we use the mathematics of probability theory in order to express the uncertainties around our model, then the inverse probability allows us to infer unknown unknowns. That means learning from data and making predictions, the core essence of machine learning and of vital importance for health informatics. So you see, these unknown unknowns, this is very important. This is getting to know completely new insight into data. Unknown unknowns. So this is very, very important. Therefore, I have emphasized this in this slide. So what is information and why is life complex information? Think back on what we have discussed about the cell. The cell as the living blocks, the building blocks of life. And to be honest, now really honest, the question what is information is still an open question in basic research. And any definition is always important and depends on the view taken. So it is quite a difference if it is taken from the physics, from the mathematics, or the philosophical perspective. So I give just an example of a definition given by Carl Friedrich von Weizsäcker. So information is what is understood. This implies that information has both a sender and a receiver, and that we have this common understanding. And we will come back to this in our lecture about explainability, because this is what we have to ensure. This common understanding of the representations, and this means to convey information to use some properties of the physical systems. So please, so far, remember, students, information has no absolute meaning. It exists relative between two semantic levels. And this again and again implies the importance of context. And I cannot always remember you how important this contextual understanding is. But without any doubt, information is a fundamental concept in our world. And life is complex information, as we have seen before. This goes back to Schrödinger, 1944, but also to Wiener, 1946. So many systems, for example, in the quantum world, do not obey to this view of information, which we have now. But the quantum world is different, and we are here dealing with the living cell, so the life, for example. And this is an interplay, and I would say a delicate interplay of energy, entropy, information, and this essential function of living beings can go back to this processing of information, this consumption and preservation and duplication of information. So this is really very important to know. And I'm very proud that informatics is the science of information. So to be honest, computer science is a little bit misleading because we deal with information and our profession is to provide the best possible information for making good decisions. So information science would capture it better, but it is computer science, as you know. But this is just a too far digression, and we will continue now. So why is the work of Bayes, Price, Laplace so important for us? Remember Thomas Bayes, Price, his friend, and Pierre-Simon de Laplace, the grand mathematicians. Why is this so important for us? Because we have this posterior probability, we have this likelihood, and we have this prior probability, and we can then divide by the evidence, the marginal likelihood. And this is a sum over space of alternative hypotheses, so this can be very difficult, of course, but this is the essence. The essence is that we have, I go back one, so the essence is this inverse probability. And as I have mentioned before, or as to be more correct, as Zubin Garamani has mentioned, to infer these unknown unknowns. So this is really about the essence of knowledge discovery, of discovery of new insight into completely unseen. So this is very important. So this is really the essence of learning, to infer these unknowns, to learn from this data, to make predictions into the future. And this is, of course, of relevance in the medical informatics domain. Okay, so probability, information, entropy, the correspondence. So information is the reduction of uncertainty. This is for us now important in this lecture and in the whole course. So if something is 100% certain, its uncertainty is 0. The uncertainty is maximal if all choices are equal probable. So this is the identical independent data distribution assumption. And the uncertainty as information sums up for independent source, just as a statement. And this brings us now to entropy as a measure for disorder. This is fascinating because entropy has many different definitions and applications, and originally in statistical physics and most often it is used as a measure for disorder. And in information theory, in our field, entropy can be used as a measure for the uncertainty in a data set. And this is really very important. So a quick demonstration how useful this can be. So you have, for example, low entropy, low complexity. You have medium entropy, high complexity, and you have then high entropy and low complexity. So you see, this has to do with the error of time. And there are a lot of different ways of measuring and what are the origins of entropy. Going back to the origins, Bernoulli maybe is to mention as the origin. So the origin may be found in the work of Jacob Bernoulli. He described the principle of insufficient reason. So we are ignorant of the ways an event can occur, and the event will occur equally likely in any way. And then this was maybe the inspirement for Thomas Bayes, 1763, and then of course later on Pierre-Simon de Laplace, actually 1774. And this was then carried out by Jeffreys and Cox, and they solidified it into Bayesian statistics. So this is called now Bayesian statistics. And if we look at the physical side, so this was more the mathematical side, and if we look at the physical side, then we have a completely similar development from Maxwell, Boltzmann, Gibbs in statistical modeling of problems in physics. And this was the inspirement for Claude Elwood Shannon. And Shannon, as you all know, laid the foundation for the information theory. And from here, and he was influenced also by Burson and by Fisher. And he developed the entropy methods, or some of the entropy methods, and also the concept of generalized entropy. So this is really very, very interesting. And now we will deal with some entropy methods in the next slide. So here you see some entropy methods, for example, Jane's maximum entropy. This is used to estimate unknown parameters of a multinomial discrete choice problem. Then you have this topological entropy. So this was introduced by Adler et al. With the purpose to introduce the notion of entropy as an invariant for continuous mappings. So in a topological dynamical system. Then we have this from Pincus, the famous approximate entropy. I will show you an example from our own work. And this leads to Richmond, the sample entropy. Then on the other hand, we have this graph entropy, Mowshowitz, mean entropy also, minimum entropy, which was then developed further by Posner, the famous Posner. And then we have from Rubinstein the cross entropy. And then the generalized entropy is was further developed by Rényi as the Rényi entropy and by the Zallis entropy. So this is just to be complete on this side. So what can we measure with entropy? You see, this is one of our students here. Don't worry, he got ECTS credits and money, of course. So with entropy measures, we can measure changes of information and therefore beautifully can locate anomalies over time or irregularities, which are very examples. For example, here, monitoring body movements along with vital parameters during sleep. This provides medically an important medical information regarding the general health, and we can so detect trends, for example, in studies and discover illnesses, including, for example, hypertension. Hypertension is often underestimated in our society. And this seemingly simple data, so this is only what you see here is from one night period, this demonstrates nicely the complexity and the boundaries of standard methods, for example, fast Fourier transformation for discovering knowledge. And what we want to know here is to find deviations, for example, or dissimilarities. And here, for example, we used entropic methods and how this works. So we have a given data set, for example, xn, where the capital N here, this is the capital N, is the number of the data points in this equation. Then we form an m-dimensional vector. Then we measure the distance now between every component. That means the maximum absolute difference between their scalar components. This is done here. And then we look, so to say, in which dimension is the biggest difference. And as a result, we get the approximate entropy. And if there is no difference, we have zero relative entropy. This is here, h of mr, where m is the run length. This is here, the run length. And r is the tolerance window. So we assume that m is equal to r, and the up n, the approximate entropy, could also be written as h tilde mr. So this is the approximate entropy of mr, where m is the run length and r is the tolerance window, and CRMI measures within the tolerance R the regularity of patterns similar to a given window with length m. This is here. And finally we increase the dimension to m plus 1. This is done here. and repeat the steps before and get as a result the approximate entropy. And now you would ask, why is this interesting? Because this gives us a good insight, and in this slide we can see the plot of the normalized, and here a good example why normalized. Here you see how important normalization is actually. So we can see the plot of normalized approximate entropy for each of the episodes and the median across all these episodes. And from this figure we can see that the entropy is a minimum where we have no alterations, and the entropy is increasing when having irregularities. And if we have no difference, we get zero entropy. And you see what we can see here. Yeah. So what is the main advantage of entropy measures? Especially over longer periods, you can quickly explore about anomalies. So you can easily detect by displaying the measured relative AppN. And what can we learn from this experiment? So approximate entropy is unaffected by noise. This is very important. It can be applied to complex time series. And this brings us now to one particular interesting approach, which is cross entropy and Kullberg-Leibler divergence, KL divergence. So what is the difference between entropy, Kullberg-Leibler divergence, and the mutual information? So entropy is the measure for the uncertainty of random variables, as we have seen before. The Kullberg-Leibler divergence is comparing two distributions. This is very, very important. And mutual information is the measuring the correlation of two random variables. So this is the famous paper by Solomon Kullberg and Richard Leibler on information and sufficiency. And if you have some silence, you can read this in peace and quiet at your home. The essence is that we should remember Shannon entropy. So please remember what we have learned so far. So information to reduction of uncertainty. And the question remains, of course, open. How do we measure uncertainty? So we can, for example, use Shannon entropy and conditional entropy, for example. So the double integral here is not complicated. This is just adding and adding again. And this is then expressed in this equation. And the question is now, when do we need the Kullberg-Leibler divergence? When x is continuous, the Shannon entropy is known as differential entropy. And if we have separate probability distributions of p of x and q of x over the same random variable, we can measure how different these two distributions are by using Kullberg-Leibler divergence. And this is just the applications. You'll see here the theta parameter again. So the Kullberg-Leibler divergence is very important, is used to measure the distance between two distributions. So this is the essence what you should get to know now. And what is important to note when using Kullberg-Leibler divergence, so while we have learned that Bayes' theorem is symmetrical, if you can remember, Kullberg-Leibler is not symmetrical. So this can cause troubles if we ignore this fact. So this is just that you remember this. So in summary, why do we use entropy measures generally or at all? So what can we learn from this? This is very robust against noise, and you will see robustness is one of the essence in all of our machine learning. Humans are very robust against noise, and machine learning algorithms, our algorithms are very, very, let's say, sensible to even to small noises. We will come back to this later on. And approximate entropy can be applied to complex time series, and time is very important. We have a good replication, and remember, replication is very important. It is finite for stochastic noisy composite processes, and the values correspond directly to irregularities. So this is very good for detecting anomalies, and this is exactly what we often need in the medical domain to find out and detect anomalies over a large amount of data where you cannot look, a medical doctor cannot look at a data set of 1 week to detect 1 microsecond of an anomaly. Okay? So, this brings us now to knowledge representation, and the question is, how can we represent knowledge in a machine? So this is really a difficult topic, and knowledge modeling is a process of creating a computer interpretable model of knowledge or a standard specification about a kind of process. And the resulting knowledge model can then be computer interpretable when it is expressed in a knowledge representation language. And we will now learn how we can do that. So again, what is the difference between data, information, knowledge? Quickly rehearsed. Data, our outside world. Inside world, we have these 3 filters. We have the incoming signals, the symbols, they represent physical values, attribute to parameters, so sensory input. We have then the information as an ordered sequence of data, for example, in an alphabet. Think on Shannon. This is what humans can perceive technically. And then we have these mental models where we check if this hypothesis which we generate fits into our mental model. And then we have these feedback loops, and you have data, we have information and knowledge. So, and knowledge is a set of expectations. Okay, so again, biomedical informatics, the interdisciplinary field that studies and pursues the effective use of biomedical data, information and knowledge for scientific problem solving and decision making motivated by efforts to improve human health. I emphasize this again because this is beautifully describing what it is about. So, medical knowledge, and this is a very old paper from Jan van Bemmel and Musen, Mark Musen. We have one big question: where does the ground truth come from? So data, here you see data are the physical entities at the lowest abstraction level. These are generated by a patient, for example, patient data, a biological process, for example, the omics data we have mentioned so far. So according to Bemmel and Musen, data contain no meaning, and now you have to interpret this. This interpretation has been done by human intelligence, so interpretation of data by a clinician, a medical doctor. So here the human comes into play. So information is derived by interpretation of data by the clinician, and knowledge is now obtained by inductive reasoning with previously interpreted data. And this can be collected from many similar patients or processes, which is added to the so-called body of knowledge in medicine. This is the explicit knowledge, and there the ground truth come from. So this is basically in the heads of the medical people, or in the books, so to say. So this is the knowledge used for the interpretation of other data and to gain implicit knowledge which guides the clinician in taking further action. So you see, this is the medical knowledge definitions. But now back to data again. So the problem is we do not know which facts are true, which facts are false. So we are living in an uncertain world, and logical representations are based on facts about the world, so true or false. These facts can be combined with logical operations and or, for example, and logical inference is based on certainty. And this is a big problem. This is now a big problem because this was actually the cause of the AI winter, and I will quickly review the AI winter later on. So because the problem is we do not know which facts are really true and which are false. We are living in an uncertain world. So that means the medical domain is not deterministic, and generalization is very hard, and our sensors are noisy, the models are not perfect, the algorithms are not perfect, the data are not perfect. So this is really very, very difficult, and I showed this slide because this is the book of Janes, and he says probability theory, the logic of science. And you see, this is a discrepancy in itself, because probability theory and logic. And this is a good argument that probability theory is the real logic of science. So what are examples for famous knowledge representations? This is just an overview site. So this is, to be honest, an excellent table, and it shows you a good overview on different viewpoints. And here, for example, you have the McCulloch bits, the foundation for deep learning. Hubel and Wiesel, this is a printing error here. This is the connectionist world in biology. Then we have the mathematical logic with Aristotle, Descartes, Boole, and inferences can be new expressions from old ones. And you have here the statistics world and the economics world with von Neumann, for example, also here. And so you can say knowledge representation in this way is a set of ontological commitments, and we will learn about this now. So what does formalization versus expressivity mean? So we have on the x-axis we have this formalization, and on the y-axis we have this expressivity. And in this slide we see a hierarchy of ontologies regarding formalization and expressivity. And you see these typical dictionaries here, data dictionaries, for example, on the left down corner. First order logic is on the far right up corner. So you see, this is a good overview actually about this, what is called now the way to ontologies. So what do you need for developing clinical decision support systems? So medical environments have enormous complexities and poses high demands on medical professionals. And here we see an example of a traditional modeling approach for medical reasoning as a basis for developing decision support systems. So this was a long strategic goal. Such models may be faithful to what it is known about biomedical knowledge, but they have serious limitations, of course, for human problem solving, especially in unanticipated situations. So this example shows the physiological factors and relations affecting, for example, mean arterial blood pressure. So this is about hypertension. You see here mean arterial blood pressure, hypertension. The history of deep learning is particularly interesting for us because it shows us a little bit about the dark age, the AI winter, if you look here. So, and this, we need this beautifully image for clarification of some terms. So symbolic artificial intelligence is the term for the collection of all methods in artificial intelligence research that are based on high-level symbolic human-readable representations of problems. That means logic and search. So symbolic AI was the dominant paradigm of AI research from the mid-1950s, so from the very beginning until the late 1980s. And this was called good, or is now called GoFi. This is good old-fashioned artificial intelligence, and this actually was the start in 1957 with the perceptron. And the approach is based on the assumption that many aspects of intelligence can be achieved by manipulation of symbols, and this was actually an assumption defined as the physical symbol systems hypothesis by Alan Newell and Herbert Simon in the middle of 1960s. And one very popular form of symbolic AI was expert systems, and they used a network of production rules, and production rules connect symbols in a relationship similar to an if-then statement. And actually the system, the expert system processes the rules to make deductions and to determine what additional information it needs. So for example, what question to ask and what what human readable symbols to use. And actually this is a very good example for early explainability, because all these methods of good old-fashioned artificial intelligence were inherently interpretable, so they were retraceable and they had this explainability. And through the success of deep learning, and now you see here, for example, we start deep learning in 1986 with this multi-layered perceptron backpropagation. This was the same time when Judea Pearl published his famous book. And then we have the success, the grand success from, let's say, in the last 10 years. So where these deep neural networks were very powerful, and we had much success with this. So this is just that you have an idea what happened in the last decades, and what makes probabilistic learning so successful, and what are the advantages of this good old-fashioned artificial intelligence. And maybe the next decade will show a little bit of an bridging these two worlds. Maybe, we don't know what will happen, but maybe. Okay, so why were early decision support system no success? So here you see the timeline. So in 1965, Feigenbaum, Lederberg, Buchanan began the work on Dendral, and then it was the doctoral thesis of Shortleaf, these mycin systems, and from mycin this was actually the father of all future decision support systems, and it was no success because it was not accepted in the clinical domain. So it was not a success, but it was the inspiration for many other systems. And yeah, it is a really interesting evolution. But you see, in 1980 it stops, and then we have this deep AI winter where no research was made on these systems. So what is the connection of early expert systems with current AI? So Dendral, this is actually Dendral, was known to computational chemists who have incorporated many aspects, many parts of their own structures. And you have here the inference engine at the very core with the knowledge base, new knowledge and modifications, for example. And here actually you have an own module. This is this image is not recent. This is 1975, and you see explanations and analysis. You see also the user interface, and you see also descriptions of new case and advice and explanations back, and these human AI interfaces. So this is 1975. So this is really very amazing. And this explanation based, and here this overall structure. So actually this is a good example for a solution in explainable AI. The user could receive advice and explanations on demand. The problem, and this was the question in the last slide also, why were they not successful? Because of their very limited application area. So they couldn't learn from new data, they couldn't be transferred in other domains. So this was very limited to very narrow fields. In very narrow fields they were successful. But of course the medical field is not a narrow field. So how did an expert system work? So this is also a original image from Shortleaf and Buchanan. So you have a database. And so actually this is mycin, this is describing mycin. You have the database with general factual knowledge, and we will see how this was stored. You have this rule interpreter. Then you have the production rules. This is judgmental knowledge about the domain. This was called static knowledge here. And then you have this dynamic knowledge. This is facts about the problem entered by user. And these are then the deductions made by the system itself. And you see also here consultative advice, explanations, so explanation capability. So actually it's a beautiful example for what is now called explainable AI. So you see this grand goal of having explanations for the underlying facts was a long sought goal for a long time. Okay, what was the certainty factor in the Mycin system? So this Shortleaf was aware of the problems involved with classical logic, and actually Shortleaf introduced the certainty factor. So Mycin is a rule-based expert system which is used for therapy planning for patients with bacterial infections. It works with this called goal-oriented strategy, Rückwärtsverkettung in German. And to every rule and every entry a certainty factor, CF, is assigned, which is between 0 and 1. And two measures are derived, a measure of belief and a measure of disbelief. And the certainty factor is then just the measure of belief minus the measure of disbelief. And if the certainty factor is positive, that means if more evidence is given for a hypothesis, otherwise the certainty factor is negative. That means if the certainty factor of an hypothesis is +1, the hypothesis is for 100% true, and the opposite, if it is -1, the hypothesis is 100% false. And of course you can imagine what happens if the certainty factor is 0. So there's either no evidence regarding the hypothesis, or the supporting evidence is equally balanced, and this suggests that the hypothesis is not true or might not be true. So Mycin's hypotheses are statements regarding values of clinical parameters for the various nodes in the context tree. And you see, this is very, very interesting. But let us look at an original example from this time at the next slide. So this is an original slide. So you see hypothesis 1, the identity of organism 1 is Streptococcus. Hypothesis 2, patient 1 is febrile. And hypothesis 3, the name of patient is John Jones. And now we have the certainty factors out as outcome of the machine, of the Mycin. So the certainty factor for hypothesis 1, given the evidence, is 0.8. So that means that there is strongly suggestive evidence that the identity of organism 1 is Streptococcus. And then we have the certainty factor of hypothesis 2, given the evidence, that is minus 0.3. There's weakly suggestive evidence that patient 1 is not febrile. And then the certainty factor of hypothesis 3, given the evidence, is +1. So it is definite that the name of patient 1 is John Jones. So this is Mycin example makes the use of the certainty factor clear. But now a few words on ontologies from our medical data science perspective. So when we talk about standardization, we immediately touch ontologies. So in computer science, an ontologies represents formal knowledge as a set of concepts within a strictly limited, and I have to emphasize, within a strictly limited domain. So this is very narrowly on one domain, and of course on domain, and of course the relationships between those concepts. So it is similar to what we have seen before, because this can be used for domain modeling. Okay, so the most important aspect which we will learn is that an ontology provides a standardized, and standardized means shareable vocabulary, which can then be used to model a domain. So this is quite useful. Okay, so what happens at a very simple example? What happens if you put the word Jaguar into a search engine? Yeah. So if you put the word Jaguar in the search engine, you will get different results. Of course, a typical von Neumann machine will know it's a sports car, but it can also be the animal or a tractor. For example, there's a company named Jaguar which produces tractors. So is it a sports car or tractor then? Or is it a aircraft? So you see, what Jaguar is it then? So without any context, the word Jaguar is useless. So we need meta information. So this meta information is information about the context. What do we know? In which context is the word Jaguar relevant for us? And who created the first ontology, or who is the father of ontology? Of course, as many others, Aristotle. And he defined an ontology as a theory of reality. This is, of course, from a philosophical perspective, but it is a conceptualization of what exists, and this is useful for artificial intelligence, of course. Here, in practice, an ontology consists of categories of individuals organized in taxonomies and connected by various relationships. So, and this is also the reason why a graph structure is often used for representing ontologies, and graphs are so important. You will see graphs are so useful, and here you see the substance, material, immaterial, body, animate, inanimate, living, blah blah blah, and so on. And this was first described in this porphyry tree. So this is definitely described, and this was a tree structure. This is the first decision tree, so to say. Okay. And in this slide, we see a classic definition. So Aristotle attempted to classify the things in the world, so where it is employed to describe the existence of beings in the world. So artificial intelligence knowledge engineering deals also with reasoning about models of the world. And therefore, AI researchers adopted the term ontology to describe what can be computationally represented of the world within a computer program, an algorithm, or whatever. And this is now the definition, the classic definition. This goes back to Studer and Benjamins and Fensel in 1998, knowledge engineering. And ontology is a formal, explicit specification of a shared conceptualization. And this is really very useful, and because other relations reflect the domain semantics and formalize the terminology in this particular domain, and the terminology again contain the terms and their definitions and usage in a specific context. You have heard now context so often, and this is a very good example for it. So a conceptualization refers to an abstract model of some phenomenon in the world by having identified the relevant concepts of that phenomenon. And explicit means that the type of concepts used and the constraints on their use are explicitly defined. And this is defined. This is very important. So this is not vague. This is well defined, and this is the big advantage of such an ontology, because it is formal, it is explicit, and this can be used in a von Neumann machine. Okay, where are ontologies used today? For example, in the Nexo ontology here, the ontology is shown as a tree with nodes indicating terms and edges, indicating hierarchical relations between the terms, and the node sizes indicate the number of genes assigned to a specific term, for example. And the node colors here represent the degree of correspondence in a term in this gene ontology as determined by ontology alignment, and with high-level alignments labeled here, as you see, and insets show the hierarchical organization identified, for example, here by the ribosome and the axon cytoskeleton. So this is a part of the network extracted ontology, so Nexo, called network extracted ontologies. And this is a hierarchical ontology of genes, so cellular components, processes derived from large genomic data sets. You see, this is shown as a tree with nodes indicating terms and edge indicating the hierarchical relations between these terms. So this is really a very good example for using on the usefulness of an ontology. So gene ontology, you have heard the term already. So gene ontology is a controlled vocabulary of terms to represent biology in a structured way. And the terms are subdivided into three distinct ontologies that represent different biological aspects. So one is the molecular function, the biological process, and the cellular component. And these ontologies are non-redundant, and they share a common space of identifiers and a well-specified syntax. So this is really very useful for us. So what is the difference then between an ontology and a terminology? So let us review. An ontology is defined as a theory of the reality in philosophy, like Aristotle, or it is a conceptualization of what exists in artificial intelligence. And in practice, ontologies consists of categories of entities organized in taxonomies, and they can be connected by relationships. And already Aristotle attempted to classify all things in the world, and consequently researchers nowadays adopted the term ontology to describe what can be computationally represented of the world within machine learning. Language, software, algorithms, blah, blah, blah. Okay, so remember please, an ontology is a formal explicit specification of a shared conceptualization. Okay, so ontology, structured description of a domain in form of concepts and relations. The is-a relation provides a taxonomic skeleton, and other relation reflects the domain semantics. So these are the formalizations, the terminology in the domain, and terminology is defined as terms, definition, and usage in the specific context. And I have highlighted for you context here. So the knowledge base is then defined as an instance classification and the concept classification, and the classification provides the domain terminology. So what are the conditions an ontology may satisfy? So this is the work of Zank and Bodenreiter, and an ontology is composed of at least one of such taxonomy and may comprise several distinct taxonomies. And the concepts across taxonomies do not stand in a taxonomic relations. So concepts in an ontology represent categories of things existing in reality or abstraction generated for classification purposes. And of course, each category or abstraction is represented exactly by one concept. Okay, you can see that here. And you can read here what an ontology may satisfy. Okay, what is a semantic relationship? So this figure presents an example from cognitive science. Here you see cognitive function, language, memory, vision. So here you have the concept. So this is the concept of cognitive function. Then you have this is-a relationship. This is the fat arrow here. And this is part of relationship. So for example, cerebral hemisphere is part of frontal lobe and temporal lobe and so on. And then you have this transversal relationship. This is this light gray arrow. Picture naming is a transversal relationship to language, and temporal lobe is a transversal relationship to memory. And you see here, verbal agility is a relationship to cognitive test and so on. So the knowledge about the brain domain, this is anatomy functional, this is like an anatomy functional ontology, is expressed through semantic relationships between the concepts of the three ontologies here. And this can be used for discovery of relationships between cognitive function and anatomical regions of the brain. And this has been done for a long time. And this is very well known in cognitive science. Okay, so what are typical medical ontologies? This is now important for you. In this slide, we see some biomedical ontologies, including the scope, the number of entities, concepts, and distribution of number of terms per entity, and the additional version names. So maybe this is updated. So SNOMED CD is very, very important. CD is short for clinical terms. We will come back to this later on. And for example, the most largest ontology is the UMLS, Unified Medical Language System Metathesaurus, which contains 1.4 million concepts at this time. So, and this is increasing because this project is still running. So, what notations of ontologies do we use? So, graph notations, for example, most of all, semantic networks or topic maps. This is an ISO standard already. Unified modeling language, as you know it from software engineering, is used for the notation. Then the resource description framework, RDF, of course. Then we have logic-based notations, for example, description logics, a lot of them. Here only mentioned a few, for example, OIL. OIL is short for Ontology Interchange Language, or TAML. TAML is agent, originated with D for DARPA, so DARPA Agent Markup Language. So it comes from DARPA. And OWL. OWL, for example, is the Web Ontology Language, so Ontology Web Language. Then you have rules, for example, Rule ML or LP and Prolog. Then first order logic, of course, conceptual graphs, or syntactically higher order logics, for example, LPs, to name one. And non-classical logics, for example, logic or non-mon or modalities. And then we have these probabilistic and also previously these fuzzy terms, for example. So fuzzy ontologies allow the modeling of real-world environments using fuzzy sets. And this mathematical environment and linguistic modeling, fuzzy ontologies became really very useful when the information that is worked with is imprecise. So, how does a graphical notation look like? So, in this slide, we see the beautiful conversation of a graphical notation. So, here you see the mapping process and some of the mapping results. So, this is explained here. And yeah, we can go on. So, what is the purpose of the web ontology language? OWL. So, DL is short for description logic. And you see, for example, here, this is the concept inclusion, so speak all C1 are C2. So subclass, for example, alga, all alga are plants, all plants are organisms, you see. And this is the concept equivalence, so speak C1 is equivalent to C2. So you see, cancer is equivalent to neoplastic processes. So how do you pronounce all these math expressions? There's a beautiful resource, the Handbook for Spoken Mathematics, which I can recommend. Also helpful, there's a list of mathematical symbols with the expressions, so how do you pronounce it? And of course, MathML. MathML is a really good facilitation for using and reusing of mathematical and scientific content on the web. And, for example, used for computer algebra systems and voice synthesis. So this can be used to encode both the presentation of a mathematical notation for high-quality visual display, as well as for mathematical content and for applications where the semantics plays a role. So spoken mathematics. This is very interesting and relevant. This is a side note, a digression, so to say. Yeah? But digressions can be useful. Umwege erhöhen die Ortskenntnis. So, what are these ontological class constructions? For example, this is the intersection or conjunction of concepts, so speak C1 and Cn, so anatomical abnormality and pathological function, for example. Or here, universal restrictions, so speak all P successors are in C, so all values from this is in the other. So, for example, all co-occurs with plant. Or existential restrictions, speak an A and P successor that exists in C. So here, for example, and co-occurs with animal. So, when talking about standardizations, we have touched ontologies. So, in computer science, an ontology represents formal knowledge as a set of concepts. And within, and I have to emphasize it again and again, the concepts are defined in a strictly limited domain. This is the big disadvantage. So if you ask for the disadvantage of ontology, it works only in a strictly limited domain. And in similar, there is the concept of medical classifications, and this goes back to Linne. This was in Carolus, in Latin, Carolus Linnaeus, and this was the first work about medical classification. So this was a taxonomy, and here the original publication. So this is one of the early nosological efforts. So nosology is the science about medical terminology, so about names in biology, in medicine. And this was, for example, grouped together in classification. So what is classification generally? It is a process in which ideas and objects are recognized, differentiated and understood, and this is the semantical part. So a classification system is an approach to accomplishing classification. And actually this goes back to further roots in taxonomy, for example, medical plant illustrations in old Egyptian wall paintings. This goes back to 1500 before Christ. And this is really very interesting that already these early paintings show that these societies valued and communicated the use of different species and therefore had a basic taxonomy already at this time. Okay, so what medical classification systems do we know? So since the classification of Carl von Linne in 1735, approximately 100 plus various classifications are in use. We only mention here a few for you, for your convenience. So the most important is the International Classification of Diseases, the ICD. So, for example, the novel coronavirus, the COVID-19, was taken on March 18, 2020 into the ICD code. And this is in effect from April 1, 2020, so quite recent. And the coding is U07.1. So this is the actual classification of disease of this COVID-19. This is known generally as corona. Okay, then the next is the SNOMED. So SNOMED, nothing to do with snow. It is instead an abbreviation for systematized nomenclature of medicine. Then we have these medical subjects headings, the MESH. Then we have this foundational model of anatomy, FMA. And we have already heard about this gene ontology, GO. And one of the largest, or maybe the largest, it's still underway, project of NHI, Unified Medical Language System, UMLS. And there is a subset of it. This is logical observation identifiers, names and codes. This is LOINC or LOINC. And from the National Cancer Institute, there's a thesaurus which is called NCI. Okay, so what is the International Classification of Disease? Let's go a little bit back. So this is the website of the ICD. And we go to the history. So how did the International Classification of Disease evolve? So it started with 1629 with the London Bills of Mortality. And this is the oldest classification of illnesses, so to say. Then William Farr in London is one of the founders of medical statistics. He developed a list of causes of death and a list of diseases. And 1893, this was von Jacques Berthelot in Paris, a list of causes of death. And from this, in 1900, the International Statistical Institute, what we know now as ISI, accepts Berthelot lists. And in 1938, we have already the 5th edition of it. In 1948, it was adopted by the World Health Organization. And in 1965, we had the 8th edition, ICD-8. In 1989, the ICD-10. And 2015, around 2015, ICD-11. And yeah, you can look up the homepage of the HWO for more details on it. So what is SNOMED then? So nothing to do with snow, but SNOMED, and SNOMED was founded as a systematized nomenclature of medical clinical terms, and it covers diseases, clinical findings and procedures. And this was interesting. This was originally developed by the College of American Pathologists, and the ownership of this SNOMED was then transferred to a new public body called the International Health Terminology Standards Development Organizations in 2006. And in 1997, we had this logical observation identifiers names and codes integrated into SNOMED. In 2000, we had this SNOMED RT, research terms, and the clinical terms in 2002. So CD means clinical terms. And this is the handbook. So you have a good documentation about it. And in July 2009, it contained over 388,000 concepts and 1.2 million descriptions and 1.4 million of relationships. And this is, of course, constantly updated. So how does, for example, hypertension look in SNOMED? So here we see two examples, a SNOMED representation for increased blood pressure and a SNOMED representation for decreased blood pressure. So a big issue in clinical information systems is the distinction between observables and findings. And there exists no universal consensus on the distinction. So the term observable generally refers to an aspects of the patient that can be quantified or qualified. So for example, blood pressure or skin color or body mass index and so on. And the finding, on the other hand, usually refers to something which is either present or absent. So possibly with additional qualifications, for example, diabetes or fractures and so on, and or the state of some observables such as increased blood pressures, which we see here. Okay, so type A makes it clear the finding of increased blood pressure implies a finding of abnormal blood pressure, and this interprets the observable entity blood pressure. And the fact that the finding of an increased blood pressure qualifies the blood pressure as abnormally high as opposed to abnormally low is not reflected at all in this expression. So this is a common phenomenon. So this has to be taken into account. Okay, what is MESH? So MESH thesaurus is produced by the National Library of Medicine, NLM, since 1960, so very early. And it is used for cataloging documents and related media as an index to search these documents in a database, and is part of the meta thesaurus of the Unified Medical Language System, UMLS. So the thesaurus originates from keyword lists of the Index Medicus. This is today Medline. And the MESH thesaurus is polyhierarchic. That means every concept can occur multiple times. It consists of 3 parts: the MESH tree structures, the MESH annotated alphabetic list, and the permuted MESH. Okay, and here the example, the 16 trees in the MESH include anatomy, organisms, diseases, chemicals and drugs, analytical, diagnostic, therapeutic techniques and equipment, psychiatry, psychology, biological sciences, natural sciences, anthropology, technology, humanities, information science, named groups, healthcare, publication characteristics, and geographicals. So how does this mesh hierarchy look like? So this is an example for the mesh hierarchy for the heading hypertension. So you see it here, diseases, it's cardiovascular diseases, vascular diseases, hypertension. And this is how it looks originally in the mass description database of the NLM. And of course, there is a national library. This is, you can see this mesh descriptors. They are arranged in both an alphabetic and a hierarchical structure. And at the most general level of the hierarchical structure, there are very broad headings such as anatomy, for example, and so on. So how does mesh look in an interactive tree visualization? So there was a nice example. So this is a really very nice example of a possibility of visualization of such structures. We will discuss this later on. So the idea of such an approach is that an end user has an idea of the overall structure. So first the general overview and then select parts of it. And this example is based on a tree map from Schneiderman 1992. And these are arbitrary trees and shown with a two-dimensional space-filling representation. And with such a tree map, two additional aspects can be displayed besides the thesaurus structure. One hand, you see it is represented by the size of the partitions, and the other hand is the color. And the hierarchy is visualized through the nesting of the areas. And the color of the different areas is used to represent the result of the different measures introduced above. So this is really a nice example. Okay, what is UMLS, Unified Medical Language System? So Unified Medical Language System is a set of files and software that brings together many health and biomedical vocabularies and standards to enable interoperability between computer systems. That was the original goals. And therefore UMLS can be used, this is openly free to everybody. And you can use it for developing applications, for example, for electronic health records, classification tools, dictionaries, language translations. And actually, this was running now for 30 years. And this was taken by the Obamacare. And it's really challenging work to classify all this and take it together. So this is really a multi-million budget necessary each year to let this project run. Okay, so the meta-thesaurus forms the base of the UMLS and comprise over 1 million biomedical concepts and over 5 million concept names. So this is really huge. And they stem all from this over 100 incorporated controlled vocabularies and classification systems around the world. And some examples are the incorporated controlled are ICD-10, as we have heard before, the MESH, the SNOMED, CD, and so on. Yes. So what subdomains does UMLS meta-thesaurus integrate? So in this slide we see the integration of various other terminologies, as we have mentioned before. You see here the GO, genome annotations, the MESH, the OMIM, genetic knowledge bases, the clinical repositories via SNOMED, and so on. So how does an example of proteins and diseases in UMLS look? So this is just an example of neurofibromatosis in an autosomal dominant disease characterized by tumors called schwannomas involving the acoustic nerve. And this is just one example, so an arbitrarily taken example. So concluding remark. Yeah, progress in machine learning is driven by the explosion of the availability of big data and low-cost computation. So this is actually very important. And in this famous paper in Science from Jordan and Mitchell, they emphasized that machine learning is the most increasing field in computer science. But what they also mentioned there, and therefore I have included it here in this presentation, is that they say health is amongst the biggest challenges for machine learning. And to be really very absolute timely, this is a blog from March 25, so very recent, March 25, 2020 by Mike Thomas, just to provide a few examples on how promising and versatile machine learning in health informatics is. This is just for your future motivation. I say thank you very much. Please stay healthy, stay at home, fight Corona. Thank you, and we see again or hear again after Easter break. Enjoy.